So while you're doing that, so let me welcome everyone to this week's Theoretical and Computational Biophysics seminar. So today we are hosting Dr. Marilia Barros from Bristol-Myers Squibb, a sort of a known pharmaceutical company who is involved in research and development of new drugs and compounds. So before I introduce our guests, I would like to just remind everyone that you're all promoted to be panelists, which gives you the option of turning on your microphone and your video on your own, so you can actually do that whenever you need to interact with the speaker. We encourage everyone to have the cameras on, so that way the speaker can see you all the time, but please turn off your mics in the beginning of the seminar to avoid any background noise to interfere with the seminar. If you have any questions, the guest has graciously agreed to be interrupted during the seminar, so you can actually turn on your mic and your camera and wait for the opportunity for a pause or something like that, and then you can actually ask your question of the speaker. At the end, we will have another opportunity for interaction with the speaker as well. So with that, it's my great pleasure to introduce Dr. Marilia Barros. So Dr. Barros is actually a biophysicist and an experimental biophysicist with expertise in biomolecular interactions, who has actually worked in drug development areas. She received actually her PhD in physics from Carnegie Mellon University. And during that period, actually, she worked on one of the subjects that is of great interest in my group, in our group here in Urbana. She studied lipid-protein interaction and the structure of proteins inside the membranes. Then during this period, she was also a guest researcher at NIST Center for Neutron Research, where she conducted and helped users actually to perform neutron scattering experiments. Then after that, she moves to Sloan Kettering Center, a famous cancer research in New York, as a postdoc. And then she was actually involved in research in compounds and mechanisms that are involved in cancer and Alzheimer's disease. Before she joined Bristol-Myers Squibb 2 years ago, where she is now a research investigator, and she's involved in actually development of biologics, which are the sort of probably most important wave of new drugs that, including antibodies or other protein-based drugs that are used in the market, probably top 10. If you look at the top 10 most expensive, most profitable compounds in the world, most of them are actually biologics these days. So the seminar is going to be a sort of a career kind of pathway type of interaction. So she's kind enough to let us know, especially the students, what they need to do and what they need to know if they're interested in industry and want to sort of follow a career path there. So with that, thank you very much again, Maria, for being here, and the floor is yours, please.
Thank you. Can you guys hear me okay?
Yes.
Okay, great. So thanks a lot for the kind introduction. I'm very happy to be here today, like sharing a little bit about my transition as a biophysicist from academia to industry. So I think first half of this seminar, I'll be exploring, talking about like my path to industry. Then I'm gonna like share a little bit of my story at BMS about the company as a whole, and also share a bit with you about the work that we do in drug product development, developing new drug biologics for our patients. So, okay, so I just want to set up some of the expectations, but I think Emma said it well. Feel free to interrupt me at any time, at any point that you have any questions. And also, as mentioned, this will be less than, not as much of a technical seminar, but will be more focused on career pathway and like an informative session about industry and how it is to be a biophysicist in pharmaceutical industry. Okay, so having said that, let me get started with let's see if I can move the slides. Yes, with my path to industry. So as you probably noticed at this point, I have an accent, so I'm Brazilian. So I did my undergrad in Brazil, and then I moved to the US to do my PhD. I moved to Carnegie Mellon to do my PhD there, to the physics department, and I joined a biophysics group at CMU. In this biophysics group, they were focusing their research on membrane-related processes, and some of the membrane-related biological processes that I studied were viral capsid formation and also translocation of proteins through different types of membranes. All of the work that I did was experimental. So I had to build these artificial lipid membranes and at different compositions of lipids and study the interactions of proteins with these artificial lipid membranes. And for that, I used a bunch of different biophysical techniques, including surface plasma resonance to look at protein interaction, nature and strength of interaction of this protein with these different lipid components. I also use a lot of neutron reflectometry to look at orientation, structure, orientation, I think I said that already, depths of insertion of these proteins into different bilayers compositions and also at different environmental conditions. So in this work of neutron scattering actually cannot be done obviously in just a regular lab. So there are only 2 facilities in the US that allows you to make this type of experiments. One of these places is NIST and the Neutron Center for Neutron Research in Maryland, and the other one is Oak Ridge. So one of my PhD advisors, he was actually based at NIST. So I had a lot of opportunities to go there throughout my PhD to do neutron scattering experiments. And then in the last year of my PhD, I had the great opportunity to go to NIST and be there for a year to finish my PhD from NIST and be a guest researcher over there. So as a guest researcher at NIST, I spent a lot of time like designing experiments, helping facility users to design their experiments, to collect their data, to analyze the data. I was really like part of the team, team of scientists, and it was really like a great time and opportunity to be there, like interacting with such great scientists and doing great science. And actually, I think the best picture that represents this time that I spent at NIST is actually this one, which is a picture of the guide hall. That's where we basically we collect all the neutron data, and I spent a lot of like late nights and early mornings there, like changing samples and analyzing my data. But anyways, it was like a great opportunity, like as I said, to interact with scientists of different fields and to make connections as well, right? So during this time at NIST, so as I said, I was finishing my thesis and my PhD over there. When I was close to the end, I had to come back to Carnegie Mellon and to defend my thesis, and I finally had to make the decision, okay, where should I go next? What is the next step that I want to take my career to? Right, and but for us, I think there should be like some international students in the audience as well. So it's not just about where's the next step, but where's the next step that can actually sponsor our stay in the US, right? So there is an extra layer of complexity associated with being international and doing your PhD in a country that's not yours. So, and on top of all of it, for myself, I had like a time constraint as well, because I think my funding was suddenly like gone, and I had to finish my PhD like with 6 months earlier than what I was initially planning. So and then I said, okay, so I had this time constraint, I had the visa constraint. It's like, and where do I want to go next, right? So I kind of had to take this decision in a short amount of time, and I didn't have much time to explore what I really wanted to do. I ended up going to a postdoc at Memorial Sloan Kettering Center. So, okay. Yes, I went to a postdoc at Memorial Sloan Kettering Center. I applied to industry jobs at this time, and I applied also to academic jobs at this time when I was finishing my PhD. And I always had this question: what should which kind of work I want to be doing in my next step, either if it's academia or if it's in industry, right? I knew that I wanted to expand my skills outside of the skills that I built throughout my PhD. I don't want to be doing the same thing, right? So in this postdoc at the Memorial Sloan Kettering Center, I had the opportunity to lead and start the biophysics research within a lab. So it was like a very positive experience overall, because I didn't have to worry about funding or money. I had like all the money and resources to build the biophysics research within this lab. So they were looking for somebody with biophysics expertise to help them understand the molecular mechanisms of this protein that is involved both in Alzheimer's disease and also in different cancer pathways. So I was also studying proteins, but a more complex protein. This time was a transmembrane protein. And I was also studying how this protein would behave in different lipidic environments, let's say. But I was like, also, at the same time, I was expanding my set of skills in the lab. So I wasn't doing surface plasma resonance or neutron reflectometry. I was like using different techniques. I did a lot of like fluorescence, high resolution fluorescence microscopy. And also I did a lot of atomic force microscopy. And actually that's the picture that I have to represent the time that I spent as a postdoc at Memorial Sloan Kettering Center. I spent a lot of time in the basement of the building, myself and the AFM instruments, just looking at protein distribution across different lipid membranes and lipid microdomains, and how that distribution of protein to different lipids to different lipid environments would affect activity of this protein towards different substrates. So I spent a lot of time in the basement doing this type of experiments, but it was a great experience just to build something from scratch that was never developed in this lab before. And also, besides like having the opportunity to develop my my skills, analytical skills in the lab, I had the opportunity to develop my soft skills as well, because I had to do a lot of collaboration, establish a lot of collaborations with different universities, with different professors, with different labs, because we didn't have any biophysics expertise, right? And I had to look for that in other places as well, so I could establish something in the lab that I was in. So
can I ask you a question, Maria? So
yeah.
here, so actually, I was just wondering, did you work with John Nagel, by the way? At
John Nagel?
No.
No, I worked with Matthias.
Oh, okay.
Yeah.
So anyway, my main question, which might be actually something that our students might be also thinking, is, so at Carnegie Mellon, you were really a biophysicist, just doing neutron scattering.
Yeah.
How did you get into Sloan Kettering, where we know it's mostly about molecular biology, cancer research? And with you are admitting that you didn't even know the techniques that you were going to work with. So how did you convince them to find a position there?
Yeah, that's true. It's like even after I moved to Sloan Kettering, I realized, okay, I'm the only kind in here, right? So most of the people, as you said, cell biologists, they work with animals. So there is some biochemists as well, but not much biophysics happening at all. I think I really convinced this professor, because he had a collaboration with another professor at CUNY, also in New York City. And this professor was more like a chemical engineer, but he was very interested in lipid membranes as well, and how proteins would behave in different lipid environments. And I think this other professor kind of convinced my advisor, my postdoc advisor, to say we need somebody with this type of expertise in this lab, because actually the lab that I joined had expertise from cells to animals to all of the stages, but nothing about biophysics.
Right.
So I think he wanted to create this opportunity in his lab as well. And I think we kind of met at the right time for both of us.
Sure.
Yeah. So yeah, it was very very surprising for me as well. But I mean, when I went there for interview, everything was very promising. He was really like wanted to invest time and money like in building this biophysics research there. So as you said, I didn't know any of the techniques that I was going to use, but I think neither him knew which techniques I was going to use either. So it was up to me basically to find out what were like the best tools to investigate that particular problem that I had in hand. So I had the expertise of dealing with proteins. I had the expertise of like working with like proteins and lipids interaction. I had the expertise of building lipid bilayers, but not looking at those lipid bilayers using atomic force microscopy or using fluorescence. But I think that's something that neither him nor I like knew that I was going to be spending time working on. So it's like it was more like it came from the needs of the project. So and then I looked for people with the expertise to, hey, I need your help to learn how to do this, how to manipulate this instrument. And then I was adapting, and I got to do the research that I needed to. Yeah, so it was very the first year and a half was very very good. I mean, I just spent 2 years there, right? But after this point, after this project was, let's say, being close to completion, there was, then I started working on projects that were more like molecular biology related. And that was something that I wasn't very happy. So that's why I decided, okay, I need either to move to another postdoc or to look for my next step, or I need to go to industry and explore how research looks like in industry. So I got to a point that the work that I was doing as a postdoc was mostly wasn't as much as biophysics as it was in the beginning, right? And I said, no, that's not what I studied for. So I'm a biophysicist. I want to keep being a biophysicist. If there is no space for biophysics in this lab, it's time for me to do something else. So that's how I said, okay, I know what I want to do. I know what I want to do. So I have time now to look for the right job and to do the right move. And then I started like targeting my searches for biophysics job in industry, not necessarily pharmaceutical industry, but biophysics positions in industry. So, and I ended up like interviewing for different pharmaceutical companies, for non-pharmaceutical companies, and I ended up like being here as a research investigator at Bristol-Myers Squibb. I joined the company 2 years ago. And as a research investigator here, my main job is basically to provide biophysical and analytical characterization of the different drug products that we have in development. And I have to do this characterization from early stage development until the product beyond commercialization. So there's a lot of support of formulation development and formulation strategies to make sure that this drug product is stable and can be delivered to the patients in a safe and in efficient ways. So I do a lot of biophysics work, and right now I'm really happy with the work that I'm doing. Yeah. So that's how I ended up moving from the postdoc to industry, because I wasn't happy with the type of work that I was doing anymore. So it was very exciting in the beginning, but then I said, okay, this is not where I wanted to take my career to. So it's time to go to the next step, and that's how I got here into Bristol-Myers Squibb. Does anybody have any questions? Because then I'm going to transition to more informative session about BMS and about drug product development. So if you have any questions, now is a good time to ask. if you have any questions about the transition, or if you have any questions in general.
No, it seems that people are fine. We can continue, please.
Okay.
Yes, I guess people might be more interested in opportunities, what's out there for them rather. Please continue.
Yeah, I'll go. I'll proceed. Okay. So, okay, this will be a more informative session about BMS, so you know more about the company and how biophysicists fit in within BMS. So the mission of BMS is basically to discover, develop, and deliver innovative medicines to help patients live better life. And I think that the good thing about BMS is that they want to do that through science, through innovation, and through technology, right? And me being a scientist, this is like very important that the company that I work for has that value in mind. They really like invest a lot of money in producing medicines that are based on science and technology and innovation. So having said that, this is like how a quick glance of Bristol-Myers Squibb. So we are a biopharmaceutical company. As we see, there is a lot of money being made every year. And I think these are our largest selling products. But I think the most important message of this slide is basically that we invest a lot of money in research and development. And again, as a scientist, that's what I want to see, right? I want to see investment in research and development, and that's the way that we're going to deliver safer and efficient drug products to the patients. So Bristol-Myers has like a global presence in more than 50 countries around the globe. Most of our product development sites are actually here in New Jersey. My site is, the site that I work on is based in New Jersey as well. I'm in New Brunswick. So recently we acquired a Celgene. So we got some discovery sites and some other development sites also in Boston and some in the West Coast as well. But as I said, most of the product development functions, they happen here in New Jersey. Okay, so we have a very strong, diverse and product portfolio. So in the last 12 years, our company had delivered like 16 new products. And basically, this is like a very good mark for a company, because you know that most of these products, they take 10 years or more to be approved by FDA. So currently we have like 97 active programs in development that goes from preclinical to phase 3 development. So this is just an overview about BMS, so you know a little bit more about the company. And then I'm going to dig a little bit deeper in the product development or drug product development department at BMS. So within BMS, we have like 4 main area focus, which is oncology, immunoscience, cardiovascular, fibrotic diseases. We also have now a virology franchise. We also been working on COVID mAb. Probably we're behind the game, the race, since other companies already have their drugs being approved for emergency use. But yeah, we're working on that sector as well. In terms of drug modalities, we work with small molecules and we work with biologics. Mostly monoclonal antibodies, but there is also bispecifics, antibody drug conjugates, lipid nanoparticles. We now, because of acquisition of Celgene, we work with gene therapy and cell therapy as well. So there is a very broad and diverse portfolio of different products that we offer to the market, right? So I think here is the important part. What do we do as part of drug product development, right? We have to transform the API or drug substance or active pharmaceutical ingredient into drug product. So here within BMS, the divisions of work, let's say, is not small molecule versus biologics or large molecules. The work is mostly divided by route of administration, So if the product is going to be delivered, orally delivered to the patients, there is one specific group, which is the oral product development group, that's going to work with this type of drug substances, and it's going to make tablets, pills. If the drug is going to be delivered to the patients through injections or through IV bags, then this work is going to be done by a different group. That's the sterile product development group, and that's the group that I'm part of. So I'm part of the group that works on liquid formulations, and basically we develop these liquid formulations that can be antibodies. As I mentioned before, it can be lipid nanoparticles, and we have to develop those drugs from bench scale until it goes to the commercial plant. So we have to support the drug development throughout like very different stages of drug development, and that's basically what I'm showing here. So to bring a drug substance from concept to the market, it takes a long time. As I mentioned before, it takes 10 years or more. And the drug product development team basically works from this stage, from preclinical until FDA approval. Sometimes even after FDA approval, we need to provide some support as well. But see, we work with the same molecule throughout the whole stage of drug product development until the product is finally commercialized into the market. So, and what is interesting here, only one every like 3,000 or 10,000 molecules that are synthesized or discovered, they ever reach FDA approval. So there's a lot of trials that don't even reach half of way of drug product development. So, and here finally we have some of the functions and activities that you can do as a biophysicist within drug product development. So there is a lot of material characterization, analytical characterization, biophysical characterization. I'm part of the analytical group that also does biophysical characterization of the drug product. There is also the formulation team. There's a packaging team and a device team. And basically there are biophysicists throughout all of these different areas. There is also a drug delivery team. So and there are, as I mentioned, there is biophysicists working throughout this whole range of different departments that we have within drug product development. So again, there is a lot. I think maybe for this audience, there is a lot of people working with modeling as well. I'm not one of them. I work a lot with in the interface with the modeling team, because we need a lot of support from them sometimes to understand what's happening with our molecules. So I don't do the simulations myself. I don't do the theoretical work, but there is a whole team behind us that work together with us to provide that information about our molecules. So, yeah, again, this is just a redundant slides, just a repetition from the last one, right? So biophysics basically can fit throughout the whole stage of drug development. And it's through discovery, in development, and also in manufacturing. There is a lot of interfaces between these different areas, and you can find biophysicists in all of these different areas. There's a lot. I think most of the biophysics employees are in discovery, but again, you can find a lot of biophysicists all over at all of those different areas and departments. So here is more a distribution of who we are at drug product development. As you can see, 2/3 of us are PhDs, but we also have the other third is like master degrees and also bachelors. I think the company is investing more in having people with different backgrounds, different education levels, because that brings different perspectives to the company, and people with different backgrounds, they see problems in different ways, right? And by major, you can see that there is a heavy presence of chemical engineers, chemists, and pharmacists. But I think this is a lot of how the company used to look like in the past. I think now the other majors are taking over a bigger part of this pie. So as I said, there is an increased presence of other majors in the company. At least that's the trend that I've been seeing now that I'm part of BMS. I can see that the new hires, they have a more varied background than not just chemical engineers, chemists, or pharmacists. So there's mathematicians, physicists, computer science. So any major you would like. So, and the other thing that I want to mention is that a lot of people think that, or have the misconception that the work that is done in the industry is not as intellectual challenging as the work that is done in academia. And I think, at least for me, the experience has been different. Of course, our main job is not publishing papers. Our main job is to support the portfolio and the product development of the molecules that we have in-house. But there is a lot of science that's done to back up the decisions that we make when we are developing our drug products. And some of those scientific work is worth sharing in the scientific community. So there is a lot of publication that's being done within the drug product development at BMS. So, and I don't want to make this like a journal club or anything. I just want to like give you like a flavor of which type of work is being done. And I try to find examples of work that's done also that has a combined approach of experiments and modeling as well. In these works that I'm going to show here, they were done either like internally at BMS or in collaboration with different universities. I just want to give you a flavor of which kind of research is done within drug product development and how we use biophysics to understand our drug product better, and how we can use that to provide better formulation of the drug products that we need to deliver to our patients. So in this particular paper, this was a collaboration that was done between BMS and University of Delaware. We've been collaborating with them for a long time now. Even before I joined BMS, this collaboration was still going on. So basically, this work was to predict PPIs, which are protein-protein interactions of very high concentrated antibody solutions using diluted solution data and also using coarse-grained molecular models. So basically, why we are interested in protein-protein interactions. Protein-protein interactions, they give us a very good idea of, or very good insights about colloidal stability of our solutions. And we have to make one of our responsibilities is to make sure that the drug product is colloidally stable. We have to make sure it's thermally stable. We have to make sure it's chemically stable, and we also have to make sure it's colloidally stable as well. All right. So, but then if you want to make drug products at very high concentration, that makes our life much more difficult, because at very high concentration, the intermolecular distance between the particles are much smaller, so they are much more prone to interact with each other, right? And if the particles are more prone to interact with each other, it means that they have a higher probability of maybe aggregating or transient or having transient interactions with each other, which may lead to undesired drug products quality attributes. For example, it can aggregate, it can have high viscosity, it can precipitate, it can have opalescence, it can have a liquid-liquid phase separation, and those are things that we don't want to have in our drug product. So we have to somehow control these protein-protein interactions at very high concentrations. So the idea here was basically to, and sometimes it's very hard to control and to measure those protein-protein interactions at very high concentration because they require a lot of material. And sometimes we don't have enough material at high concentration to do the experimental measurements. So the idea is to measure the material at low concentration and use molecular modeling to simulate or to predict the concentration, to predict the interactions, the protein-protein interactions at high concentrations. So that was the whole idea of this collaboration, right? But if you go and dig deeper in this paper, you're going to see there's a lot of challenges related with the modeling of the antibodies, right? I mean, how come only 12 beads can properly give a complete picture of an antibody, right? So obviously this is a very coarse interpretation of how antibodies are. It doesn't capture the complete picture of the antibody. So there is a lot of issues with this modeling, but there is other follow-up papers where they refine the model, where using more beads, and they get a better accuracy on the predictions of protein-protein interactions at high concentration. So this is one of the works that one of the routine type of analysis that we have to do here. We have to measure protein-protein interactions. We have to, because those measurements will tell us if the protein is prone to aggregate or not, right? So this is like very crucial information because we have to make sure that the drug product formulation is stable. So this is just one example of the type of work that can be done within drug product development that will help us to make decisions regarding the formulation of the drug product that we wanted to deliver to the patient and to the market. So this is another example of the type of work that's being done here, that was done here. So this investigation was done within BMS. There was no collaboration with any other external partner. So during one of our routine analysis that we have to do, we found out there was some anomalous peak in a short variant profile. And basically, we know that we are supposed to have just one peak. That's the standard, right? So we see one more extra main peak there. So where is this peak coming from? What is it like? Does this mean that the product is unstable? Is this related with some kind of product instability? So we have to dig deeper and understand where this other peak is coming from, right? And then to do that, again, we ask help of our modeling colleagues, and they're gonna model build homology models for the antibodies, and they're gonna do some molecular dynamic simulations. And basically they saw that there is this loop that's extra long and extra flexible for this particular protein. And then this loop, depending on the pH, can be extended or can be folded. So there is a pH-dependent conformational dependence of this loop. Whenever the pH is neutral, the loop is extended. If the pH is below 7, then the loop is folded. And basically, this opening and folding of this loop exposes this tryptophan. So, expose these tryptophans and also changes the pKa of two residues, two nearby residues, and the exposure of these residues basically change the whole net charge of the protein. So that's where the two peaks comes from. So whenever the loop is extended, the protein has one net charge. Whenever the loop is closed, the protein has a different net charge. And whenever you do the assay to look at the charge variant profiles, you can pick it up the two conformations of the loop. One is extended and when it's closed. Again, that helps us to make an informed decision about what is the best formulation for this type of protein. Do we want to lock? We want to have a pH that's gonna lock the loop into the folded format, or we wanted to have the loop extended. So we want to have a pH for the formulation that actually have the loop in a closed state. Because when it is on the open state, then there is more hydrophobic residues exposed, which induces more aggregation. So I just want to give you a flavor which type of work we do here and how this type of research can help us to make informed decisions about formulation development of our drug products. So all the research that we do is related to the portfolio. It's not that we are researching something that's completely out of nowhere, that's not related, that's not applicable to our molecules. Everything that we do is like with the purpose of improving the formulation and improving the development of our drug products. So this is the type of work that we as biophysicists do here in drug product development. And I think to finalize, I just want to say that there is a lot of opportunities within drug product development, internally and externally. So there is a lot of opportunities also to do research collaboration with academic partners. So where we can write internal grant proposals within BMS. These grant proposals are approved by a scientific advisory board. They give us money. We go outside, we partner with academia and we develop research such as we did with the University of Delaware. And also for individual people, we have like different positions, full-time positions, temporary positions. We have internships opportunities, we have postdoc opportunities, we have co-ops opportunities. We actually have 2 open positions now in the formulation development team. One here in New Brunswick, another one in Summit, also in New Jersey, and they're looking for biophysicists. So I think that's all that I had for today. Just want to thank you, everyone, and please ask me questions.
I can start because I'm sure people are more interested. So these opportunities, like the internship opportunities for our students, is this something that you announce on a yearly basis somewhere people can look up, or how should the students find out about this opportunity? Are these things on your website somewhere?
Yes, exactly. So we're trying to get better at this. But there is information on the website, just bms.com, and then you're gonna find opportunities about internships, co-ops. But we are currently developing a website for drug product development, in particular, just focus on drug product development, where we're gonna be posting on a monthly basis about the new opportunities that are within drug product development. So, but that's something that's not out there yet, but hopefully by beginning of next year, this should be available.
So do you have restrictions for students? Do they have to be citizens or green card?
Usually for internships, yes. That's what I've seen, that they usually require citizenship. But I think there is also an alternative of using CPT, I believe, for some of the international students. And I think that's also, that may be possible. Yeah.
I see. So Jack, you can go ahead and ask your question. So the forum is over. Thank you very much for the nice presentation. So now this is open. So I think we're gonna keep just this same webinar moving. and use it for the question and answer and interaction with them. Jack, you submitted something. Jack Stubenbol, if I pronounce it correctly.
Okay, so Jack is asking what phase of development does the majority of the funding in R&D go towards? That's a good question. I'm not sure if I know how to what's the answer for this question, since I'm like a very lab-based person, so I'm not sure where the money goes. But I think there is a lot of investment on the initial phases, like discovering, trying to like understand like which synthesizing molecules, and trying to understand which molecules are the right molecules to the right target. So I think there is a lot of investment in the beginning phases, of development.
I see. Mia, you can actually turn on your microphone and ask your question. Mia Tekawa.
Yeah, hi.
Hi.
So I was just wondering, because I think you mentioned earlier that you, on the East Coast, they focus mostly on the product development, but I'm from the West Coast. So like those locations, I think in Southern California, do you know like what the focus would be there, if any of the product development goes on there or not?
Yeah, so there is a lot of discovery going on in our California side in Redwood City, and there I think because of the merger with Celgene, there is another site in San Diego as well, and in this side of San Diego there is some product development going on there as well, but it's more towards drug delivery systems.
Mm-hmm.
Got it. Okay, thank you.
Yeah.
So in terms of interaction with universities, Maria, so now obviously, I mean, I'm sure many groups are interested. So do you guys go out and sort of approach the groups that you know, or do you have announcements about opportunities in certain areas? So let's say if I'm interested in interacting with your company, how should I go about this?
That's a good question. Usually we go finding the opportunities at the academia, because we know what are the gaps that we have within the company. And for example, we don't have that expertise internally. So, and then I know somebody that has that expertise in academia, and then I contact that person and say, hey, do you want to collaborate in this subject? So this happened recently to me. So last year I applied for an internal grant proposal, so that I wanted to explore high-speed AFM capabilities, because I think that's going to be useful to understand better like the flexibility of the antibodies, and understand the antibodies in a molecular level in general. So I knew somebody at Cornell that had like the expertise on high-speed AFM, and asked him if he was interested in joining this proposal, and from there we started. But I think we are open to maybe receiving emails saying, hey, I have this idea, I think your company may benefit of this. Well, let's talk. I think we are open to that too, but there is no specific channel for like attracting the academic community to come to us. So it's usually more the other way around. But it's actually a good point. I'm going to talk to my higher ups and mention to them that's something that would be interesting to have as well.
Right. Was it Simon Schering at Cornell?
Yes, exactly.
This time my guess was correct. The last one was close, but okay, cool. Well, no, so he's great. Wonderful.
Yeah, he's great. So I knew him from my time at Memorial Sloan Kettering Center. So right. I said, okay, I know somebody that can help us with this technique. So that's how I got to him.
Absolutely. No, I guess I'm sure there are many people who are interested. So we do a lot of membrane protein simulations, and we can offer our kind of expertise, but without knowing actually what are the systems or what are the needs. So
definitely, I totally understand. It's like, and we are investing a lot of, we want to really develop our computational and expand our computational like team. So we're definitely looking into outside expertise to bring that into the company. I think that's something that's a very hot topic, I can tell you right now, within BMS. So that's something that they want to spend some money on.
So we have Lauren.
Yeah, I was wondering, like, what experiences do you recommend for students to do during their undergraduate career to prepare for like a career in drug development, like courses and like research outside of class?
So I'll tell you a little bit about my experience. I didn't take, I think networking is the best, to be honest. And I mean, doing research in labs, right? In any lab, it's just getting, if you know that you want to be like a bench scientist, for example, just getting exposure to science, right, in general. So if you just join a lab and like do some projects, some research for like 6 months, 1 year, or longer, if you enjoy the work that you've been doing there, because I think that's a great way of learning like different techniques. In terms of courses, I'm not sure. I mean, if you're more theoretical or more computational, that maybe having a course in specific, I don't know, language or in specific subject, that may be more relevant. But for us experimentalists, I feel that like getting hands-on on like projects, the same goes for theoretical and computational people as well. It's like just working on, like joining a lab, working on projects, and learning from like postdocs and grad students, because that's where I think that's where you're gonna benefit the most. That's something that you can put in your resume, right? So you start that's when we select for some internships. Most of the students are undergrads, and we see the lack of they put something that's not related with science in their resumes, right? So we are not interested in knowing that you I don't know, that you I don't know, you are a pharmacist, or that you sold something. We don't care in drug product development. We want to know like which kind of like scientific experience you had during your undergrad. Did you join a lab and you had you work on a project there? So this is the type of things that we're looking for. That I think that's my advice. In network, always just participate in this type of connect with different people, right? Because you never know who is going to be the next person to offer you an opportunity.
Absolutely. And the needs actually of the company might change, you know, the projects they might be interested in. So it's always a good idea to be connected and to know more about the nature of needs and projects that the company is interested in.
Yeah.
Jack?
So I'm a computer science major interested in going into bioengineering and drug development. So do you know what role computer science plays at BMS?
Computer scientists. Let me try. I'm trying to think like if I know anybody from computer science. I know some people, because I think it's the way you have to think is like this. Don't put yourself in a box, right? So because you're a computer science, doesn't mean that you cannot take like different roles within the company, right? I know people, for example, that have background in civil engineering, that they are working in devices. So we would talk like, how does that happen, you know? So sometimes you just interested in how the people in the thought process, in the critical thinking, if you're a good scientist, or how you think about problems in general. So don't put yourself in a box. I'm computer science, I can just do I don't know, computer science related type of things. There is a lot of data mining happening. There is a lot of they're using a lot of artificial intelligence doing different projects. So I mean, I don't know the details of somebody that has like a very strong, I don't know anybody, I can't think of anybody that has like a strong computer science background and where they work in drug product development, but I can definitely look into that for you if you're interested. You can connect it with me on LinkedIn and I can give you more information on that. But yeah, I'm sure there is plenty of computer scientists around to help us.
So do you have, Maria, do you have some job fairs or where people from BMS physically come to the campus and try to recruit people?
So we try to go to career fairs that the universities offer. So we try to be present on those, and we're trying to increase our presence on those. So I think this year there was a career fair at Carnegie Mellon and another one at Johns Hopkins. We were there. We had some presence in both of them. We are planning to organize symposium every year where we're gonna bring students to Bristol-Myers Squibb, where they can present their work and they can also learn from what's the work that we do here as well. We're planning to do that this year, but because of pandemic, we had to postpone all the events for next year. But yeah, we're definitely working on bringing people in-house, and so they can know the facilities that we have, know more closely the type of work that we've been doing. So.
I see. Because what I hear from the students, there seems to be a lot of interest, but they just don't know if their expertise would match what the company needs right now for a particular development line, kind of.
Yeah, my advice is like, don't be shy. It's like whenever you're applying for jobs in general in industry, you're never gonna check all the boxes, right, for the skills that are requiring. So as I said, I think some positions they are really like looking for those specific skills, but I think some other positions they are more looking for are you a good scientist or not? You know, and I think, to be honest, most of the positions open in drug product development team or with the interfaces, the groups that I work more closely with, they are more interested in are you a good scientist or not, instead of do you know how to use this instrument or not, because this is more like a technical thing that everyone can learn, right, can be trained on, but they want to know if you're a good scientist or not. It's like something it's different.
So even though you almost answered the next question, I'm going to call Alice.
You pretty much answered it. Thank you.
Yeah. So when I joined, I'm a biophysicist. I joined a group that's mostly analytical group. So the work that they do is mostly chromatography, and it's mostly chromatography, that let's say. But I joined this group, but I'm doing like biophysics work in this group. So because they wanted somebody with expertise in proteins, in biomolecular interactions. So, you know, you don't need to be like you don't need to match everything. So yeah. So
I always tell my students, actually, most companies, if I had a company, I would be interested in smart students who have analytical skills and who have done research. They know how to do research.
Right.
The technical details usually you can learn later. So that's an important point actually to keep in mind for all of students. Absolutely.
Yeah, so like being on this side now, like BMS side, so I had the opportunity to work on hiring new employees as well, and I could like understand how they think about, okay, when they're hiring people, what they're looking for when they're hiring those people. And I feel that the group that I'm in and the department that I'm in, they're like more interested, as I mentioned, in those like, are you a good scientist or not, instead of like I know how to use it.
Okay, any other questions? Oh, Pepe.
Yeah, I wanted to ask you if you think that with all the home office that we are forced to do due to the coronavirus and all this mess that I think will still stay on for a little bit of time, do you think that actually pharma will opt for more computational people since they can still do the work from home with no problems? So do you think there will be more space for modeling and computational science?
I think, I mean, talking about BMS, yes, for sure. But I think that's something, I think not necessarily because of coronavirus, but because we really need more people with computational and theoretical expertise, right? Sometimes our experiments don't answer all of the questions. Sometimes the experiments are limited and cannot give all the all the answers. We need like some modeling computational work that's going to help us to give insights on what's happening here and there. I think we'll be, the pharma industry in general will be evolving to that side, but not necessarily because of coronavirus. Like we specifically at BMS received a message saying we're not going to become a work from home company. So they want people to come to work like whenever it's safe, of course. So we have even me as an experimentalist. So I still can work from home because we have a lot of data to analyze in the end of the day, right? To not just like set up the experiments, but I think the main part of the work is actually analyzing the data and interpreting those results.
Okay, good. Any other questions? Can people contact you, Maria, with questions later?
Yeah, for sure. Feel free to contact me. You can find me on LinkedIn. I can make my email. Maybe I can enter my email somewhere. I don't know.
And put it in the
chat box.
Maybe.
Okay.
Don't mind, so you don't have to. But I thought maybe there are other questions people can ask, especially students.
Yeah, I can also send the two positions open for biophysics.
Yes, actually, you can actually send it if it's a little bit difficult for you to do it right now. You can send it to me and Donna, and we can share it with all the biophysics, biochemistry students who are attending. Yeah. So that might be a better solution.
Okay, I can do that.
Yeah, great. Thank you very much again for making yourself available. So I think on your schedule, it says that we might have another session to meet, but that was meant to be actually for questions and answers and interaction with you, which we already did.
Okay.
Then I think we are good.
Okay, sounds good.
Again, it was great. And I see a lot of thank yous also in the chat box for your presentation.
Okay, thanks for the opportunity. It was great. Thank you. Thank you, everyone. Bye-bye.
Bye.